person is a researcher that's at KI in the Department of Biochemology, Tumor and Cell Biology, and he developed and published. So he has been working on deconvolution quite a lot, and he actually published a software, deconvolution software called deconvolve or deconvolve. I don't know how you say it.
Yeah, but either way, I think we understand it.
So, today is going to talk about 2D and 3D deconvolution. And then, so we will have a short talk now. And then afterwards, we will go to the workshop Zoom and we will actually do some exercises. And after that, you have one portfolio assignment, which is to deconvolve your own images. So please, Eric, thank you for being here and go for it.
All right, you can see the screen.
Yes.
Good. So I will give you a very brief, just 15 minutes, introduction to deconvolution. So my plan is just to provide you some intuitive understanding of what it is. I will try to point you to some potential problems and show some examples to motivate you. So if we were in a class, I could ask you how many used deconvolution before. I would just assume that some of you did, some of you maybe don't. And I will spend now 3 slides just to explain what it is in the quickest possible way, I think. So I will start with this game. This is called Minesweeper, and you can find it online. And if you've played it, great, because this gives really a very good intuition about what deconvolution is and what kind of problem it tries to solve. If you haven't, and this looked just strange to you, play it afterwards. I assure it will pay off in 10 minutes. So in this game, the goal is to find the hidden mines, which are red here. And each mine gives a signal or something that can be detected a bit away. And if there are several mines, their signals, they add up, just like the point spread function does for fluorescent signals. So in some cases here, it says 5. It means that there are 5 mines hidden in the vicinity, but it's nothing there. And to see the connection now between this game and deconvolution, I converted this, I took the mines, I converted it to bright pixels instead. So imagine you have a microscopy sample with these white pixels, and we have a toy microscope that has a very tiny point spread function. And I assume you know everything about point spread functions now from the course. So if we would apply this point spread function, so convolve the image with this, it would produce the same effect as looking through the objective. So it would look something like this. So something similar to a microscopy image. And here you see I marked the sources in red. So now the purpose and the goal of deconvolution is to find back the emission sources, or to say reverse the effect of the point spread function. And in this case, it's very simple. You could do it by hand. Just looking here, you can probably figure out where the true spots are, although there's some regions that are a little problematic because it's very dense. And also the algorithm that I use for deconvolution will have problems if the signals are very dense. And the real problem is more complicated because the point spread function is bigger and there is noise also and other effects. Right, so deconvolution, it's not just an image enhancement. It's not just a general deblurring. It solves a quite well-defined problem. That is what it is. Okay, but why would you like to do it? So the obvious answer will be that you like images that look better, it looks good in the paper and so on, but I think the main reason is that it has or can have dramatic effects on the quantification. And you have to bear with me a little more with my synthetic images, because here I have a synthetic 3D image where I put a lot of small emitters to the right. And on the left plane, the ones I marked with the arrow, there's a emitter that is twice as bright as all the other ones. So imagine you have a sample containing this. And just to make sure that you see that it's actually brighter on the left, if I take this image and adjust the contrast, I can do that until I only see the points to the left. Okay, but under a microscope, how does it look? So again, I take my simulation microscope and I convert it to something that looks like wide field. So here it looks very blurry because all the small emitters, their signals, they combine and enhance each other. So you see a big cloud to the right, this small dot to the left. But if we change the contrast here again to really see what is the strongest, you would actually see that the emitter that was the strongest doesn't look like the strongest anymore in the image. So if you have any quantification here where that matters, or actually if you just try to distinguish the true signals from the noise, you will be a little fooled if you don't do deconvolution, at least of the wide field. So it depends on the modality also. And how does it look in real images on real data? I will show that now. So to the left is a maximum intensity projection of a raw wide field image, and to the right, the same one after deconvolution. And if we were to detect the strongest dots here, just say we do the 1000 strongest. Here's where they sit in the original image. And I must say there are other ways to detect dots. You don't have to do by maximum intensity. So this is particularly sensitive to absolute intensities. But are these the strongest emitters? Yeah, as you heard before, probably not. So just to show you a little, this is not always intuitive. So if I take now I show the raw image in both right and left, but on the right, I've marked the locations of the dots detected from the deconvolved image. And they are in really different places, and especially they don't cluster around the bright regions. And if you're not, if you don't believe that the image to the right shows better locations than to the left, you go back, play some Minesweeper, and then look at this again, and maybe you will be convinced. So this is the major reason why you want to deconvolve, because you get a more true representation of the emission from the sample. Okay, if you're convinced you want to do it, how do you do it? It's not that tricky to do it, really. But you need to know the point spread function of the system. And here, there are three major ways to get around this. Typically, you would use a theoretical model to calculate the point spread function. And if you use the most basic ones, which you probably start with because they only require a few parameters that you can get from the scope directly. Those models are symmetric. They're both rotationally symmetric and mirror symmetric around the lateral plane. If you go for a more advanced model, it's trickier to set the parameters. It's also possible to measure the point spread function from beads or tiny emitters like RNA fish probes. And the blast, which is probably the best, but it's not that many options to find them, is to use a parametric model to fit the measurements to a model to figure out some parameters of the model. But as you know also from previously in this course, it is tricky with models because there are all these sources of aberrations, a lot of things that are not really ideal in microscopy. And just one thing is how objectives are designed. They're typically designed, they have a design case which is to look just below the cover slip. And as soon as you image things further into the sample, the PSF will change. And And even if you get everything correct, there is also the fact that the sample is optically active. Otherwise, it would be invisible to microscopy. So it actually diffracts light, it absorbs light, and so on. So this is very complicated. And to the right, I've plotted a cross section of a PSF from one of our scopes from an objective, which it really isn't that symmetric like the basic models would generate. And if you have a PSF that looks like that, you might have to work a little to get really good results, to find a good PSF model. Then it's the question which method to use. I suggest you use a linear method, a method which assumes that the measured image is a linear sum of point spread functions and some noise. And for that, there is the Richardson-Lucy algorithm, which is more or less optimal in terms of image quality. And whatever you decide to use in the end, I think you should always benchmark against an implementation of this. And now there are also neural network methods, which I think especially can be good if there's really noisy images, and also they can possibly speed up the processing. But these methods, they require training data. And it might be that even if you spend time training them, you don't get much better results than the Richardson-Lucy. But you would have to check the latest papers on that if you want to go that route. So here's the general scheme. If you want to deconvolve, start with high quality images. I mean, that's why you're here, to figure out how to get them better. Know the point spread function of the system, of the objective, and also how it performs for the particular emission wavelengths that you're detecting. Get familiar with the software. They can be very resource demanding. You have to also figure out a lot of parameters, like the number of iterations. Maybe there are options for boundary handling. Maybe it can do pre-processing for you. Maybe you have to do that before. Possibly it scales the images in some way you have to invert afterwards. And finally, be very critical to the results. And there's always the question if another microscopy modality would be better, if possible. And maybe if you deconvolve, you would need to compare with some super resolution imaging anyways, if you find something surprising to rule out that it's an artifact. And with that, I continue and I will show you some examples just before we round off. Good and bad, mostly good. Here's an example where the non-deconvolved is on the left, deconvolved on the right. It's quite good. But here's one typical artifact, at least in my images, is the string with the point inside. And this can be seen now very clearly because it's a super bright dot to begin with. But this is caused by mismatch in the point spread function given to the deconvolution program and the actual point spread function of the system. So there's a mismatch in shape here. Here's an example showing that deconvolution also works not just for point sources, also for like line structures. So here it's before deconvolution. I've zoomed in on a small region. I've circled this dark thing here and deconvolving. And it's obvious that there was a structure there that couldn't be seen before, really. Maybe if you change the contrast a lot, you would see it, but it's obvious, and these lines are more homogeneous in intensity. Right. And of course, nothing prevents you doing it also on super big images. Here's a 140, now it's almost 200 megapixel image. So before and after deconvolution, here's just shown the DAPI channel, so nuclei staining. Also here, it makes sense. But what about other modalities? So confocal. See, the confocal has much tighter point spread function, much smaller. So what can we expect in this case? In this case, the improvements will be very modest, and I'll show you here. So you could get results that look a little crisper, but also here it's maybe even too many iterations because I think I see maybe a little, at least it looks a little more spotty, and it doesn't look that calm to the eye. Maybe it's true, maybe it's not. So if you need to know, you would have to compare with super resolution. And just last, you met Hans today, I think, and here's one of his images captured with stead to the left. And also in this case, the convolution can be applied. Anytime the imaging can be described as a linear combination of point per function, it should work to some extent. So also here, it looks like it even improves the resolution of the stead. Right, so those were my examples for now. And we go to the take-home messages, which are two in this case. I start with the good. Deconvolution is great. It is very nice, especially for wide field images where the point spread function is large and there's much to do, much to improve. And the best thing of it is that it gives pixel values that better corresponds to emission intensity, so it helps in quantification. On the other hand, the convolution is annoying, and it can introduce artifacts. I mean, if you deconvolve a lot, you will find these yourself. And those artifacts, most of the cases, you will spot them. But if you have automated analysis pipelines, it might be tricky in some cases. And then, of course, this is a threshold just to get started. There are many options and many parameters. Also, it has a reputation that it takes a very long time, and it takes some time for sure, but computers are also much faster, and the most software nowadays can use also graphic cards to accelerate the processing. So it shouldn't take much longer than actually taking the image with the scope. And with that said, you will get some experience in the following workshop where you will try yourself. And I think it's about time to end now. So we will also have questions in the workshop, in the beginning of the workshop.
Okay, great.
So thank you.
Thank you. So let's go to, so thank you, Eric. And thanks to everyone to follow online. So we'll see you in a little while. We're going to go to our workshop. We meet again in a second in the other Zoom.